in my notes, I said to you guys, so the history of things is sort of this, that, an ancient, pretty old physician in Britain figured out that enzymes and genes were associated. So the first kind of rule was, one enzyme, one gene, right? And then Beadle and Tatum got it to one protein, one gene. And then as we started to understand more about protein structure, then it was, one polypeptide, one gene. So that's sort of the progression through things that there are. The whole idea of Beadle and Tatum is that they were really able to demonstrate that certain mutants lacked enzymes, so they really were taking that British doctor's, whose name is escaping me right now, something with a J, I can't remember what his name is.
Gerard? Or Jarrod?
Was it Jarvis? No. J-A what?
Jarrod.
Oh, Gerard. Oh, yeah. Gerard. Yeah. So they were able to take his conclusions and really make sense of them in a really great and sort of amazing experiment about mutants that could survive with mutant fungi that could survive with or without certain amino acids and things. But the results of their experiment was really the one gene, one enzyme hypothesis. Okay. So then as you're in this chapter, the first thing that happens, I saw on the internet that there's a picture of a dead white deer buck from someplace back east where a hunter shot him. Everybody's mad at him for shooting it. The deer was apparently kind of ancient and decrepit, and everybody's pissed off that he shot it.
Yeah, there's been a lot of people posting pictures of killing animal wildlife, and
is that right?
They just take it out.
Like kind of rare ones that.
Yeah, I think they did a bear, and then they did a lion I want to say a lion. And this girl had one of them had a modeling contract, and they dropped her.
Yeah. Oh, did they drop her?
Yeah, they dropped her.
I heard about her, the beauty contest winner or the something. Yeah. And there she was holding a dead lion or something. Anyway, okay. So moving forward here, so we go to protein synthesis, and the first thing is, what's the overview? And this is called Francis Crick's central dogma. So Francis Crick of Watson and Crick kind of got, it's DNA to RNA to protein, right?
Yeah.
And so we've been studying that as really the flow of genetic information, and we've studied it since almost day one in here. And this time in this chapter we add a nuance to it, and that nuance is in eukaryotes that we have to add in RNA processing. So it's transcription of the DNA into some sort of pre-messenger RNA that is a messenger RNA that's longer than what the ultimate messenger RNA is going to be, or an RNA that's longer than what the ultimate messenger RNA. And it has to be edited, and it has to be kind of modified. And so what you guys learn about pretty quickly here, I'm going to skip ahead a few pages, just stay on this same topic. What you learn about not too far into the future here is there's this thing called a spliceosome. And a spliceosome is this very clever array of RNA molecules, again, and some protein. And that spliceosome's job is to grab that pre-messenger RNA and to cut it up and to splice it into ultimately the shorter thing, the messenger RNA. Does that make sense?
Yeah.
And so the spliceosome does that by grabbing the pre-messenger RNA and removing these little units in there. The units are called exons and introns. And you really are tempted, you want to say keep in the introns, right? That's the obvious way to remember it, except for that's wrong. Okay? So it's throw out the introns and keep the exons. Someday I should look up the origin of that word to understand why they did it that way, because it still seems backwards to me. So that's what the spliceosome does, is it cuts out the introns and puts together the exons to give you your message, okay? And soon after this, I told you about something called alternative splicing. And so I'm really kind of zipping ahead, and I'll have to go backwards a little bit, but since it's kind of consistent on one subject. So this figure here shows you the relationship between the various exons. You know, it's got introns in it that haven't been spliced out yet, but it's showing you that pre-messenger RNA, and it's kind of saying, oh, by the way, exon number 1 that's gonna stay codes for this part of the polypeptide, this domain of the polypeptide. And exon number 2 here codes for this domain of the polypeptide. And exon number 3 is, you know, the end of that polypeptide. So this is going 1, 2, 3, just like that. But it turns out in eukaryotes, we've also got this thing called alternative splicing, where somehow natural selection figured out that this kind of useful piece of a protein could be mixed up and put in a different place, and it could make another useful protein. And so part of what's going on with us seems to be that some of our proteins are the result of this alternative splicing, and that simply means that instead of going 1, 2, 3, it's going to go 2, 1, 3, or 2, 3, 1, or something like that, right? Some sort of thing. And that's called alternative splicing. So that gives, the point of that is it gives eukaryotes a greater diversity of these biochemicals called polypeptides. Does that make sense? So alternative splicing gives, you know, increases the diversity of the biochemical polypeptides. Okay. So then, kind of going back and picking up where I left off a little bit when we were talking about the flow of genetic information, they have here a nice little, here's the DNA double helix, then here's some RNA that gets transcribed from it, and then here's the polypeptide sequence, the amino acid sequence, I should say, excuse me, based upon using this table here. And I promise you guys, I'll ask you, it'll probably be about 2 questions, but it'll, you know, 2 questions for sure, maybe more, about something that says you can do this. Okay.
What page is that?
So this is on page 329 and 330. So let's just practice for a second. So if I give you, you know, a sequence of DNA, it goes something like this.
Yeah.
This is the 3' end, this is the 5' end. Then I'm going to ask you guys to transcribe this. Okay. So to transcribe this, you're going to convert it to RNA, 5' RNA to 3', right? So just initially here, we're going to have, if we have A's, we're going to have what? An RNA.
Is it you?
A's and U's, right? So this will be U, 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 U. There's a T, so that gets an A. There's an A, so that gets a U. a C, that gets a G, that gets an A, G, G, U, U, C, 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 G, right?
Yeah.
And it's going 5' to 3', right? Now, which way does the ribosome read?
3 to 5.
5' or 5' to 3'?
3 to 5.
5 to 3.
The ribosomes read opposite of the enzymes. The enzymes go 3' to 5',
Yeah.
and the ribosome reads 5' to 3'. Okay. So that ribosome is going to kind of come along and it's going to start reading this thing. But it's not going to start reading it. It's going to ignore until it finds AUG. So AUG is going to be the start. And then there it's going to put down it's just a different color. There it's going to put the amino acid down, methionine. Okay. And then we have an AGG and a UUC. So for an AGG, we go over here. AGG, it looks like arginine. What was the next one? UUC. UUC. Phenylalanine. So we'd have arginine to phenylalanine. Right, and then we got a CCG.
So we pretty much just use the table, right?
Yeah, yeah,
Okay.
you don't memorize it.
Okay.
No, I provide you the table.
Okay.
So, CCG, CC is proline. And then I need to have some kind of stop. I need to have a UAA or something like that here,
Yeah.
which is the stop, right? Does that make sense?
Yes.
So I need you to be able to look at the DNA, figure out which way is 3' to 5'. You know, I might have you going from right to left or left to right, and then make your RNA from it, and then make your DNA from it. I mean, excuse me, you make your protein from it, but using the table. You know, you never would have to memorize that. Well, you might have to in a biochemistry class, but that seems absurd to me.
Not even like a AUG or anything?
No, so AUG you should know.
Okay.
And that the other thing you should know is that there are 3 stop codons. So UAA is one of them that you can say UAA, UAG, and UGA. Probably would impress me, but I probably won't ask you. I'll probably won't ask you a question that's going to say, "Do you know those?" Right? But AUG, you got to be able to go. Oh, I got to find the start codon, right?
Okay.
Does that make sense?
Yeah.
Okay. So let's just talk a little bit more about this table. It's organized into threes, and three bases together makes a codon or a codon, depending on how you want to pronounce that word, right? And so you need to know where codons are. So these triplets are found on the messenger RNA, right? And what's on the transfer RNA? On the transfer RNA, there's going to be the anticodons, right? So there'll be codons and anticodons, kind of like matter and antimatter. And they're gonna, you know, sort of meet up with each other. Okay, is that part okay?
Okay.
All right. So those are kind of 2 big picture things. Now it gets more specific. Now it says in the chapter, okay, let's talk about transcription first. So it's transcription, then translation to do protein synthesis, right? And so transcription, the good thing about it is RNA polymerase, that's your enzyme. That's the answer to all the transcription enzyme questions. What enzyme does this in transcription? Oh, it's RNA polymerase. What enzyme does that in transcription? Oh, it's RNA polymerase. Always the same one. Okay? So RNA polymerase is the thing that comes along, and as this picture shows, comes along and it attaches to the DNA in a place called the promoter region. Okay, and the promoter region just means start. So there's this start place. And that just helps the enzyme to attach. And then the enzyme goes about splitting that DNA double helix right down the middle, just like helicase did in replication, right? It splits it down the middle, but it doesn't just keep on going splitting. It also takes time to now start adding RNA nucleotides to one side. So here it's got the, sorry, I can't read it. Okay. So here on the bottom, it has on the bottom, this is the 3' to 5' strand here. So this RNA polymerase is reading the bottom strand, and now it's starting to replicate that bottom strand from a 5' to 3' direction, and so it's starting to build the RNA right there. And eventually that RNA kind of falls out of the, you know, after about 10 nucleotides or so, it kind of falls away from the DNA double helix. It's sort of too heavy to be there. And this thing just keeps going until it finds its termination code. Okay?
Okay.
And so then ultimately, when this thing finds the stop codon, the RNA polymerase breaks off of the DNA wraps back up into a double helix, right? And the RNA and the new polypeptide both fall away from the DNA double helix, so they just kind of float off away from they're all separated now. In reality, what typically happens is this gene's getting transcribed, and there's another RNA polymerase right behind it transcribing it, and there's another RNA polymerase transcribing it. So usually what happens is, you know, a bunch of these things are going all at once. Is that okay?
Yeah.
Okay. This box over here says, okay, now you understand that. Here's the detail. Let's just add in that about the promoter region. So it gives you some idea what a eukaryotic promoter is. And a eukaryotic promoter is a little more complicated than a prokaryotic promoter. It has this thing called a TATA box, and that's just TA, a bunch of the T's and A's nucleotides put together. And so that TATA box is also joined with certain proteins, and these proteins are just transcription factors, they're called, and these transcription factors just kind of help to orient the new enzyme to the right place, and that new RNA polymerase gets loaded by those guys and then just starts going, right? Is that okay? Okay. So that's kind of all those things. I don't think there's anything here that's earth-shattering. This is just a blow-up of what's really going on here. That all seems the same to me. That all seems like you should understand that at this point to me. This, I had mentioned to you about processing of the pre-messenger RNA, and they're kind of back to that topic here. Hi, how are we doing?
Fine.
What's happening?
So, Monday,
Yes?
are you gonna be finishing the Drosophila lab, etc., etc.? Yes, because Karen using computers, and on Wednesday you're gonna use computers.
Yeah, I'm actually gonna do Hardy Weinberg lab and show them some video and
What
stuff,
do
but
you
I'll
need
do
from us?
I just your good wishes.
Okay, I will
Okay.
be here with
Okay.
Dr. D.
Oh, so actually, here's what I potentially need on Monday, just to let you know.
Yeah.
You know, we're just rushing to try and replace Roy and we've squeezed in a committee meeting at 7:30 a.m.
On Monday.
On Monday. It goes to 9.
Uh-huh.
So
Yeah.
I assume that I'll be done in time. I mean, that I will get enough
If
feedback
not,
that I
I
can
will
just
be
walk
here.
away. But if you see that I'm not here by 9:15 or by
8:45.
8:45, if you
Open
see
the
I'm still
door,
not
let-
here yet, just open the door, let them in, and I'll be in. I'll
And
be in.
what else? Would you like me to?
No, nothing.
Nothing?
I don't
Okay.
think so.
I'll do it.
Unless I run over here and say, watch this movie. I don't know.
Okay,
But, yeah.
got it. Don't
Okay,
worry.
thank you.
Okay, I'll be here. On time.
Okay, thank you.
Thank you.
So I was saying that we've already talked about the need to process that messenger RNA. But here it's just telling you, by the way, here's some of the processing that's done. Besides taking out the introns and leaving only the exons, there's this funny thing that happens where they put on what's called a G cap. And so that G cap, I don't know if you can see this. It's got 3 phosphates on it, so it's like GTP, right? That's the G cap there. So it's a funny little thing, and that's at the 5' end. That's the part that's going to go into the ribosome first. So it must be important about attaching to the ribosome or getting the ribosome to grab in there. And then at the other end, they do a poly A tail, which just means A, right? Okay. So this processing then included eliminate the introns, a G cap, and a poly A tail. And that's like a perfect multiple choice question for me, right?
Eliminating introns, right?
Yeah, eliminate the introns. The things that should stay in don't. They get eliminated. Okay. So that's transcription. We did it. Okay.
For your question, it says explain what it means to say genetic code is redundant and unambiguous.
We got to do translation before we can get there. Okay. Let's do translation first.
Wait, so it's it puts on the G3P cap?
It puts on a GTP, right? GTP cap. Not a G3P, that's photosynthesis.
Okay.
Okay?
Yep.
Okay. So then we get to translation. And for translation, this is my favorite picture because I just don't know. It reminds me of New Year's, right? Confetti falling everywhere or something. And so, this is kind of telling you everything that's going to happen in translation. The ribosome is going to be the major player here, right? The ribosome has 2 parts to it. It's got the major fragment and the smaller subunit, the major subunit and the smaller subunit. They assemble around the transfer RNA, and it's going to jump along from the 5' end to the 3' end. It's going to jump along 3 codons at a time, right? It's going to, 3 nucleotides at a time, or 1 codon at a time. It's going to just keep sliding along there once it finds that AUG, right? So if it was like this one, A, that ribosome would just kind of keep sliding along. It would have 3 A's and then 3 A's, and it just keeps sliding along in there until it finally got to ATA, and then it would get to TAC, and that's where it would. Oh, I'm sorry, not the ribosome. The ribosome would be on the we'll do it differently. The ribosome would be on the RNA. So it'd be going along. It'd be reading 3 U's, right? U,
Yeah.
and slides again. U, different U, slides again. Another U, and then it's going to get to UUA, and then it's going to get to UAU, and finally it's going to get to AUG, and that's where it gets its orientation from. And it starts there, right? Does that make sense?
Yeah.
Okay. So then, this picture is also great because it shows you the ribosome has 3 slots to it. The E slot, the P slot, or the peptididyl slot, and the A slot. And the A slot is where the new transfer RNAs are going to come into with their anticodons, and they're going to match up to the codon. The peptide bond is going to be, or the peptide is going to be bonded to the transfer RNA that's there principally in the P slot, and the E slot's just an exit or eject kind of thing, right? It's kind of like, picture somebody shooting a rifle. It's just like exit that thing out, right? So the ribosome has the tunnel that it reads the RNA in. It has the 3 EPA slots. And up here someplace it has something called a catalytic site. And that catalytic site is an enzyme-like function where it takes and joins amino acids onto this thing making a peptide bond. So it's catalyzing peptide bonds between amino acids. Okay? So that's what the ribosome does, those three things. Okay?
Yeah.
Okay. The other thing that this picture shows you is, oh, okay, so look at what's going to happen. In the E slot, this guy's going to get ejected, a free transfer RNA. And that transfer RNA is going to go off and find a particular amino acid. You know, so whatever its anticodon is going to be key to what kind of amino acid can actually bind here. So not just any amino acid can bind, but a specific one, right? So that's happening someplace out here, and that's an enzyme requiring reaction, and it requires energy as well. All right. So here's the transfer RNA, kind of showing you a sequential sort of thing, showing you these, you know, these three little cul-de-sacs or the shamrock sort of shape. When it twists, it kind of gets this. And what's interesting about it, again, is the anticodon, the thing that's going to match up with the messenger RNA, and this other place called the amino acid attachment site, which has, in reality, has a whole bunch of proteins, some enzymes here as well. And those enzymes are going to have an active site that will allow it to hold a particular amino acid. Okay. Here's their schematic view. Anticodon at the bottom, this little fuse of the firecracker is the amino acid attachment site. The whole point of this is simply, hey, look, empty amino acids get reloaded so that they can carry empty transfer RNAs get reloaded so that they can carry amino acids. And that's an enzyme reaction, and that's a reaction that causes energy, which I've already said to you, or requires energy. Okay. Then we get to here, and we're saying, okay, well, let's see what the ribosome's really doing. So first, they try to show you a relatively realistic picture of what a ribosome looks like. I don't know if that'll ever be valuable to you in this class anyway. But it does illustrate for you the three slots. It illustrates for you that the messenger RNA goes through this area between the two subunits, really. And then, let's see, three slots goes through. Well, and then there's going to be some sort of peptide bond catalyzed there. Okay, this thing's just showing you the relationship between a codon and an anticodon. So I often ask the question, if I've got a sequence of DNA, you know, what's the anticodon here? Or something like that. Somehow I'll have a way of asking what's the anticodon. And so you have to be able to go, oh, okay, that's the thing that's complementary to the messenger RNA, right? Just remind me, I said we're having a quiz on Monday too, right?
Yeah.
Mm-hmm.
So evidence for evolution and proteomics.
Yes.
And so then here's the final kind of, here's what translation really looks like. So starting up here at the top, three slots of the ribosome, only the P slot is full. That P slot's got a small polypeptide already built on it. Here comes a new transfer RNA that's going to be somehow guided in to fill the A slot, and then we're going to have both the A slot and the P slot full. What's going to happen next is there's going to be a transfer of these materials together. So in this case, it actually looks like the P slot one is going to get cut off and added to the A slot one. So down here and you see, oh, look, A is the one who has the long polypeptide on it, and P doesn't have any peptide at all. Okay? So then spend a little more energy. The first energy, by the way, was to do what? Was to bring in that transfer RNA. Spend a little bit more energy, and what we should have happen then is, I'm sorry, I'm getting sleepy. P there, blah, blah, blah. Okay. So then what we're happening here is down here, the transfer of the, let me say this again, I messed up, I got really sleepy. So the catalytic site over here takes and removes the polypeptide and adds it on to the, for the amino acid in the A slot, so there's been a transfer of that there. Now the P slot's empty, now the ribosome slides 3 or slides a codon over, and then when it does, then that sends this guy out, that ejects this guy empty as it is, and it's going to go out and look for new amino acid. And it's going to bring in yet another transfer RNA, and that transfer RNA is going to go there. It's going to just keep going through this and going through this and going through this until there's a release factor. And that release factor comes from UAA or UAC or UAG, right? Or UG. Think about it that way. UAA, UGA, or UAG.
And that's all the stop put on, right?
That's how you get a stop, yeah. Okay, and so then this next figure shows you what happens when the release factor is there. So the release factor is basically a transfer RNA without any ability to carry a amino acid on it, and it spurs everything to fall apart. And as it falls apart, it releases then the parts of the ribosome, the messenger RNA, the release factor, and if it had a polypeptide on it, then that polypeptide would float off like this one does too. So this last figure down here is kind of showing you the whole big picture right there on what is their rough endoplasmic reticulum, and just kind of showing you what happened step by step to kind of get you to review the steps of protein synthesis. Okay? Is that okay?
Mm-hmm.
So then the last part of this chapter is about mutations. Okay. And the reason we spend time talking about mutations, of course, is because mutations are the ammunition of natural selection, right? Once we get mutations built up in a family, then there's lots of choices that can be made, and we can find who's better off in a particular environment to some degree. So in this, we first talked about point mutations. Point,
One second. I completely forget. I have a Zoom call at 9 a.m. on Monday.
oh, okay.
So I will be here. I will open the door to them like 20 minutes before.
Oh,
Right?
okay. That's fine.
Let them in.
You don't have to
Tell
come. I'll do it.
No. I must be here early morning
Oh,
because
okay.
I have another stuff to do.
Okay. So if you just leave it open, that'll be fine.
Yes, I will let them in, and I'm moving around. I will be back.
I'm pretty sure. I mean, there are only 24 applicants.
Good luck.
Yeah, 24 applicants. I'm pretty sure we're going to get through it. And he's got an hour and a half budget. I'm pretty sure we'll get through it in an hour. I know.
Unless if somebody will start talking.
Well, I'm pretty sure I can make my opinion known, and I can come here and let them do whatever.
Good luck anyway.
Okay. Thank you. You two have a terrific weekend, Anna. Okay. So this is all about mutations, and first we're going to talk about point mutations. And point mutations are just where there's a change at one nucleotide. Okay. And so they give you this classic example, one that we've already talked about a bunch, sickle cell anemia. And they're saying, look how important they can be. Here in sickle cell anemia, in the in the 8th position or so. Is that it? Is this the one that shows you? Yeah, this one doesn't show you. But it's not very far into it. That's like the 8th position or something. A single change of A and T, just actually a flip over of the A and T here is enough to have it cause, I'm pretty sure it's the 8th amino acid, maybe it's the 6th, of the hemoglobin molecule to switch from glutamate to valine. And that switch is enough to cause the hemoglobin molecules to crumple down into little bars when the metabolism gets too acidic inside the cell. And that comes about when the parasite from malaria happens. And so this one little thing, one little change, just a change of one nucleotide, switching where here it says TA and now it says AT, that little switch is enough to do something pretty dramatic, something that if you lived in a malarial area would be pretty wonderful, right? Does that make sense? Okay. And so then this next part is meant to explore these point mutations. And so we can explore these point mutations by learning first whether they are missense or nonsense. And a missense mutation is one where there's a substitution, and that substitution of the nucleotide gives you a different amino acid. Okay? A nonsense mutation is where a substitution in the nucleotide sequence creates an unexpected and unneeded stop codon. So since there are 3 possible stop codons here, that could happen, right? So in this one, the new thing it's showing you is that methionine gets put on and then it's stopped and then it's time to take the methionine off, and that's it. That just ends protein production there. There's nothing but there's nothing there. Does that make sense? Okay. And then, oh, by the way, up here, it says, there's one other kind of point mutation that's kind of important. That's called a silent mutation. And a silent mutation comes about because the stuff we already know about the genetic code. We know that the genetic code is redundant, that a codon has information where it could have any of 4 nucleotides in the first position, any of 4 nucleotides in the second position, any of 4 nucleotides in the third position, so that a codon, 4 times 4, 16 times 4 is 64. A codon has lots and lots of possibilities of what it can be. But there are only 20 amino acids, so we have this redundant 20 amino acids in living organisms. So we have this redundant system. And that redundancy is most easily seen if you look at the 3rd nucleotide of the codon. And so in this particular case that I'm pointing to, GCU, GCC, GCA, GCG, all 4 code for alanine instead of coding for different ones. And you can see that kind of redundancy everywhere. Threonine is coded for by these 3, proline coded for by these 4, etc. Does that make sense? I said 3 and I'm in 4. Boy, I am really sleepy. Sorry.
Does that mean that there's no effect on the amino acid sequence?
Yeah. So that's what a silent mutation is. A silent mutation is, oops, the DNA sequence changed, but we didn't change the amino acid sequence, so nobody's going to know by looking at the proteins. It's a silent mutation. Is that okay?
Yeah.
So then there's a second kind of mutation. So this could be caused by a substitution, right? There's a second kind of mutation that can be caused by either an insertion or a deletion. So either adding something in, so where that says UUU, if it suddenly said UUA, whatever, I've added something in, that's an insertion. And a deletion would be I can just go over and remove something. If I remove that A on AUG, that would be a fairly catastrophic deletion.
Or like if it puts like a U between a A and a G.
Yeah, or we can
Oh.
put a U between the A, we could put another U there or something like that, yeah. And so those guys, insertions and deletions, are called frame shift mutations. So the idea is, this is the reading frame here. AUG, AGG, UUC, CCG, UAA. That's the reading frame. But, you know, you could have other mutations that cause a change to occur. So, for instance, we could add, you know, something in this third codon here. We could call this one AG. Well, let's just check. If we call this one AGC, it's probably still going to be arginine, so that might not change anything. Oh, wait, I'm sorry, I'm doing substitution. Change that again. What did I have?
AGG.
We're getting so many of these. Oh, yeah. Remember that. So ARG. AG. Sorry, I'm having trouble finding it. AGA is arginine, okay, or AGG. So if this was AGG still,
That's right here.
I woke up at 3:30 in the morning just freaking out at all the stuff I have to do for you guys and other things. I have just fallen asleep now. So AGG, we're talking about a frame shift mutation. So what we mean is, well, what happens if something else gets added in here, right? Or if something gets lost.
Yeah.
So a frame shift mutation could work. I could take this A out, that gets deleted out, that would be a deletion. Now suddenly the frame is GGU, UCC, CGU, right? We get a whole different sequence.
Yeah.
Or similarly, what was this U? Did I already say U here? Do you guys remember?
A.
A. So similarly, the other possibility is we could add in another letter here. So now maybe I add in another A. Now that gives the frame here, and there, and there, and, you know, there, and that gives you a whole new sequence as well. So that's what's happening. Those are,
Frame shift.
that's a frame shift. Okay, does that seem good?
So it's always with only one base card.
No, actually, that's the thing. It could be more, so it could be 2, or it could even be 3. If it's 3, that's a special kind of frame shift where you're really just adding a whole new amino acid in there. And so your protein might be completely unaffected depending on where that amino acid is that you're adding. You're adding a whole new one, and otherwise everything's the same. If it's on the end, you know, it's kind of that amino acid that's dangling out there that nobody cares.
And that goes for both insertion and deletion?
Yep. Yep. Okay. So we've talked about transfer RNAs, we've talked about the whole process, we've talked about mutations. Is there any other mutation thing I need to mention to you? Mutations are often caused by chemicals called mutagens. That's pretty much it. That's the chapter. So I like this as a way to study because this really gives you pretty much everything. You've got to do the transcriptional modification of the, post-transcriptional modification of the messenger RNA. You've got to do all kinds of stuff to go through this. This is just a fun figure in terms of all the things that exist there. Right. Is that okay?
Yeah.
So then we smartly skipped gene regulation and the little bit of molecular technology that I was going to teach you. And we jumped on to chapter 21. No, chapter 22. Chapter 22 is about evolution. And so this is the evolutionary thought chapter. And this one's kind of important for you to read and digest because this just sort of sets the picture about evolution, although I've been talking to you about it so long now that you guys probably aren't gonna, you aren't gonna probably encounter too many things that are different than, or surprising to you, I guess might be the better way to say it. And so, first off then, we talk about some early ideas about evolution. In this figure here, it tells you about a guy named Hutton. Hutton was a geologist, a Scottish geologist, and he proposed the principle of gradualism. And gradualism was this idea that the world has changed based upon gradual, everyday sorts of processes that we see: erosion from water, erosion from wind, erosion from glaciers, etc. And then it takes a long time for it to occur. And so Hutton, and then later Lyell, when Lyell kind of grabbed onto Hutton's ideas and kind of expanded them to uniformitarianism. Hutton and Lyell were really just arguing that Earth is shaped by the regular everyday processes that we see. Nothing spectacular had to happen. No big catastrophes had to happen like the Bible said with a big flood, or as Cuvier, the Frenchman, picked up in his ideas. And so Cuvier was said, contrastingly, Cuvier, the Frenchman, said, oh, I get it. First there were these organisms, then there was a big catastrophe and they all died. Then some new organisms came into the air, then there was a big catastrophe and they all died. Then there was another group of organisms and then a big catastrophe and then they all died. And so Cuvier was kind of saying that the world is shaped by catastrophes. And Hutton was saying, you know, it's just gradually done. I mentioned Lamarck and Plato in here, so you should kind of go back and try to remember what Aristotle and Plato, what Aristotle had to say. And then we get up to the Frenchman Lamarck and eventually Darwin. And so on these pages, it just says, okay, let's take time out and talk about the fossil record and how the fossil's made. And, you know, really all I need you to know is that fossils are made when an organism dies, usually getting trapped in some kind of wet sediment that keeps it away from the oxygen, so and buries it. So first off, no bird flying overhead can come down and pick at the skeleton because it's buried away. And secondly, being buried away like that, so suddenly it might not have much in the way of fungus or bacteria that could go at it and mess it all up. And so that quick burial makes it likely to become a fossil, but not even sure at that point. And there's still lots that can happen to it. And then we get into Lamarck's hypothesis. So after you get the idea of what a fossil is, we get into Lamarck's hypothesis. And Lamarck is the Frenchman who said that things that happen in your life are going to be somehow concentrated in your sex cell producer, so gamete or ovary, sperm or ovary, depending on who you are. And that means that, according to his theory of inheritance of acquired characteristics, it means something goofy. One of the ways to interpret it is with a giraffe. And so if you remember, a giraffe has a long stretched neck, and the idea, according to Darwin, would be, oh look, some animal that had a relatively long neck must have done well in years of drought, and it passed its offspring and its alleles onto the next generations, representing those longer necks, because those longer neck guys are the ones that could do well in the drought periods. And then there's a little bit here that says, here's a demonstration of what would happen if the theory of inheritance of acquired characteristics was true, then this man's bonsai tree couldn't give rise to a large, whatever this is. Looks like it might be a redwood. No, it looks like it's a spruce. This spruce could do what it could, if by the particular theory of inheritance it could give any kind of offspring, a huge diversity of offspring. But from the kind of hypothesis that Lamarck came up with, gave us the acquired characteristics, that means that this guy, with its shortened roots and shortened leaves and trunk and all that stuff, that this guy would, if the seeds of it were taken, would only produce little bonsai forests, right? And instead, that's not true. Bonsai is an art that has to be continually kept at. That's not what happens. These guys could have seeds on them that are regular, everyday size, pine trees or spruce trees, whatever these are. That makes sense? So then it gets into Charles Darwin, and with Darwin, the place that they inevitably are going to start is his very early experience here. So it talks to you about who Darwin was, a religious guy going to study for the priesthood, or not the priesthood, the ministry, because he was so discouraged by modern surgery and all the screaming that went with it. He graduates from Cambridge, and he is given a position on the ship, the HMS Beagle, that's going to go chart the coast of South America. He's told by his professors, he's told how to collect rocks, how to collect trees, how to collect animals. He's taken copious notes. He's got crates of specimens. He sends them back at every port he can. And so there's just all this stuff is kind of rocking Europe that Darwin is sending them. Meanwhile, his 2-year trip to map the coast of South America turns into a 5-year trip around the world. And so this guy at the end of that gets back to England, where he's more or less the toast of the intelligentsia, the intellectuals. He's kind of the toast of those guys, because, you know, he'd gone on this very, very long adventure and seen all kinds of remarkable things, and he was apparently quite a good storyteller. And so they'd invite him to dinner a lot, and he'd tell stories about things that he saw. And eventually marries his cousin Emma Wedgwood, of the Wedgwood China fortune. So there's a Wedgwood China, and she was one of the primary beneficiaries of that fortune, and so she was a rich lady. And so Darwin didn't really need to work. He just spent most of his time at his house working out the problem that had so long vexed him about common ancestors, and diverting and changing from a descending with modification from those common ancestors. And then he eventually gets pressure to publish when another guy, Alfred Russel Wallace, sends him a manuscript that says, "Eureka, I figured it out. You know, I understand what's going on here." And he writes a manuscript that doesn't use the word natural selection, but it basically describes exactly Darwin's idea. Darwin, you know, has his ideas from his trip. His trip impressed upon several things. I should go back and say what they were. When he got to Chile, when he got to Brazil, the eastern side, he was immediately just astounded by the great variety of organisms that existed. And so he saw this great genetic diversity. He saw diversity within and between species, and that just wowed him. Then he got up the other side by Chile, and he saw this mountain range, and this mountain range had fossils at great depths, but since they were raised up, the fossils were there. And so at the base of the mountains, he's finding fossils, and those fossils turned out in some cases to resemble very closely organisms that he found living and running around in the mountains. And I gave you that little example of the armadillo-like thing that had a big old morning star on the end of its tail, and they undoubtedly used to beat each other's heads with it, but happened to be the size of a VW bug, and that's similarity to an armadillo. But even easier than that, you know, he would find impressions of ferns, and then he could go see the live ferns and say, "Oh my gosh, this hasn't changed hardly at all," etc. As he gets around or leaves here, the third really big observation comes when he gets to the Galapagos Island archipelago. And in that Galapagos Island archipelago, he is quickly met by the governor of the Galapagos, who's the brother of one of the politicians over here on the mainland, and gave him the job. "Yeah, you go out to the Galapagos, get away." And so Darwin hears from that guy that he can immediately tell what island the strange giant tortoise is from by looking at it. He'll know whether it was, from what particular island. And Darwin's very interested in that. As it turns out, then he doesn't really see much in the birds, but he shoots a lot of birds and brings those birds back to England. And an ornithologist later tells him, "Hey, you know what? All these birds are from the same common ancestor." And that ancestor is, probably a flock of birds that got blown off course here in some windy thing and somehow made it out here to the Galapagos and survived. And the same deal with the tortoises. They floated out and got to the right place. Okay. So diversity, fossils that look like living organisms, and then all this specialization of organisms, individuals onto different islands, and therefore a speciation event. Okay. Then where do we go? So Darwin, this note here is worth just commenting on. Natural selection is all about differential reproduction and survival, right? Those individuals who survive better, those individuals who are best adapted, have the best equipment, in other words, best songs, best feathers, best beaks, whatever, those guys that have the best are the ones who survive longest and reproduce the most, or reproduce the most, because some organisms just reproduce once, right? But they could work the other way. Darwin also said, oh, I get it. And in this paper he said, it also means that everybody's from some common ancestor, which was just one or a few cells as far as he was concerned, that got invented by a process he didn't understand. We now call it chemical evolution. But from that place, then a whole bunch of diverging lines can come, and these ones up here are the species that are still existing right now, and those guys are showing some that went extinct, and those guys went extinct, etc. Does that make sense? Okay?
Yeah.
Okay. So then, here's a modern version of a phylogenetic tree, just to show you, what can happen with these things now. This is showing you the relationship between a funny little organism called a rock hyrax, which is about this big. I saw them in Africa from quite a distance, but I saw them. They were just lying out in the sun. Oh, actually, no, I saw them close up too. That's right. They look like they'd make great pets.
They're what?
They're that small. They look like they'd make great pets. But from that guy, along came a group of animals that include the elephants and include the sea manatees, or the Sirenia. There are sea cows, I guess they're called, over in Florida. And so this gave rise to those guys, and so they have this sort of common, remarkable ancestry. And so then the rest of Darwin's book was all about, well, how do I know this is true? Why do I believe this? And so he spends time accumulating evidence. And so this chapter does, and looks at some of the evidence Darwin accumulated, and then it throws in some other stuff. One of the things it points out to you is that essay that Darwin read upon the time he got back by Thomas Malthus, where Malthus was worried about the population of the world with humans in 1799. Darwin transmitted that, or translated that, into talking about how overpopulated a local population might be, and not all of those kids are going to survive. So maybe he wanders onto a meadow and there's tons of little baby ground squirrels over there, and he knows full well by the year that those number of ground squirrels are going to get smaller and smaller and smaller. The babies are either going to move elsewhere or they're going to die. And so he asked the questions, okay, well, who survives and who dies? And his answer is, it's those ground squirrels who have the best adaptations, the best equipment. Those are the ones who survive and reproduce. And so what that means then is the population's going to become more and more and more adapted to the local environment, because you start out with this population, and the guys who had a beak like this were the ones who really survived, and all these other beaks that are whatever they are, those guys didn't survive as much. So mostly these guys survive, and most of them reproduce, and then over time that trait takes over the whole population. Okay.
Survival of the fittest.
That's where the phrase "survival of the fittest" came from. Yeah. Darwin also knew, but the environment could change, so that could reverse. Everything could reverse and stuff, and so it is a little bit confusing that way. So then he talks about various different things that are evidence for his ideas. Artificial selection is one. Here he's got this great example of, look at what happens when you take wild mustard and you select it for flowers and stems. Oh, you end up with broccoli. Look what happens when you select it for, it says selection for apical tip, but for buds on the top. So something from little buds on the top turns into cabbages. Well, look what happens when you select it for the leaves. Oh, you can make this strange vegetable called kale that's so popular, etc. Right? So this page has too many young are born because populations are relatively stable and they're all not going to survive that year, and some of them are just going to get lost. And this population says that, or this page also says that artificial selection is just a really powerful tool. And what if he imagined a process, natural selection, where who gets to breed is also really important. So that was what's going on there. Then they give you a couple of examples of camouflage behavior, or camouflage things. It's just an example of what natural selection could bring. And then they talk to you about drug resistance and pesticide resistance and such. So those were kind of boring pages. And then it gets to another one of Darwin's favorite kinds of evidence for evolution, this thing called homology, which allows him to study anatomy. And he studies this anatomy and he goes, look at this. The phalanges of a person come from the same part of this animal as the lower foot. And so he's got something curious there to deal with. Then he's got this one here, the anatomy of this one, the whale fin. It's the same anatomy that could make a bat wing. In fact, the bat wing has even the bat wing has as fewer of the phalanges and things that this guy did, and so the bat wing is just different because these guys are exploiting the air and these guys are exploiting the water, but still the idea is they came from a common ancestor. So Darwin saw this kind of evidence in all kinds of things. He saw it in embryos, that embryos from relatively different vertebrates would bear some strong resemblances, particularly at a young age. And two of the easy characteristics to point out there are the post-anal tail, the one that comes out that even humans have, but that gets reabsorbed, or the fish's pharyngeal pouches, or gill slits as they're sometimes called, the things that are there in a young fish that are going to be eventually built into gills. These are both homologies, or things that impress him, because they stretch into many different species at that embryological level. Is that okay?
Yeah.
Okay. So then, some other evidence he looked at was this idea of biogeographical behavior, or biogeographical distribution. And we talked about that in lab too, but in lab I did it more in the sense of continental drift. And the continental drift hypothesis had not yet been invented for Darwin. Darwin had no idea that was what was going on. So this kind of biogeographical distribution had rules about, say, convergent evolution. And so in convergent evolution, I gave you this picture of the sugar glider from Australia and this tree squirrel from North America called Glaucomys volans. That guy makes his living by jumping off the tops of trees and flying down into other trees, and then it gets its food out of the trees and then moves on and goes elsewhere. And the sugar glider does the exact same thing. It's remarkable. They do the same thing to make their living, except that these guys are a part of the earliest divergence in the mammals. So if you think of the tree of life in mammals, here's the early mammal that came from a reptile. That first divergence off, practically, the things that eventually give rise to the modern mammals and the rodents like that squirrel, those guys diverged off this way. And over here are the guys that are going to become marsupials, amongst other things. And so these things here at this point in the division, they don't even look like squirrels. That happens way more out here when things have divided many times. And so there's this remarkable division that says two unrelated organisms have converged onto or have merged with the same phenotype, and that's what convergent evolution is. And that amazed Darwin because it really connected phenotype to the environment. It said, hey, look, these two unrelated organisms became, essentially the same phenotype or pretty close to the same phenotype, even though they have different equipment inside of them there because they've been separated for so long. Is that okay?
Yeah.
That's a convergent evolution, right?
That's convergent evolution. Yeah. And that's sort of in contrast to adaptive radiation. So adaptive radiation is more like this, where there's some sort of common ancestor of some mammals, and what do you know? Through a series of time periods, those mammals radiate into a whole bunch of different species, and so they fork off and go in different ways. So I had the display back there with Hawaiian honey creeper birds and Darwin's finches, and then with horses, and then with the primates, eventually showing you that diverging, branching thing. That's called adaptive radiation. But those two big views of how things work are commonly pitted against each other in a series of arguments that about how things came about in different kinds of organisms. And so for you guys, I just need you to be able to compare convergent to evolution to adaptive radiation, right? Does that make sense?
Yeah.
So that was this whole chapter. And then we've just started into the next chapter. And I've told you that we're talking about microevolution. And microevolution is really going to be based on processes that biologists can measure and see, and that we think that these same processes that occur at the population level, because populations are what evolve, we think that these are the same things that gave rise to the big patterns in the animals and the fossil records, the plant animal fossil record. And so those big parts of history, that they call macroevolution, and that's actually chapter 25, and that's where Bio 4b starts. Okay, so we're building up to you guys understanding evolution, and then you're going to start to look at the diversity of particularly animals and plants and how they're organized. Animals a lot you'll do. Okay, so in this chapter there are just a few things that I think there are just a few things that I would have to get across. You guys okay? Are you falling asleep as much as me? Are you okay?
Okay.
I've never fallen asleep giving my own lecture before, so this is embarrassing. So in this case, then we're starting off first with the idea of genetic variation. So I've made this big deal that the whole point is, okay, now we've got all this genetic variation from meiosis and from random mating and from jumping genes and things, all those different things that can cause genetic variation. Now we've got this variation, and natural selection is going to come in and edit it, and it's going to say, okay, this is a really good variant, and this guy leads to an adaptation that helps this thing survive in this environment. This is a really bad one. Hell, those things are going to die out, right? That's what natural selection does. And so then the first point they want to make to you is, oh, by the way, not all genetic variation shows that kind of fidelity to genetic underlying as you think. Here's just one that happens to have both switches that are contained within the genetics here, and this is just showing you here's a kind of phenotypic variation that doesn't really match up with the genetics. So not everything's going to work that way. In this particular case, somehow these caterpillars have a switch in them that turns on based upon the kind of food they're eating. And if the food they're eating are the flowers, then they take a form that looks like the flowers. And if the food they're eating matches more the stems and some of the leaves, then they match this twig-like thing. And that's just it. You're not going to do anything with selection to change that. Okay. Then it says, oh, by the way, how do we measure genetic variation? And so it tells you, the first way was, well, let's just go out and measure the proteins and and find out how many different proteins are produced by each of the genes, how many alleles there are. And then they talked about having average heterozygosity, or average number of alleles that are average number of genes that have alleles at them that are variable rather than just fixed. And so that was one way. And then more recently, we've gone to DNA sequencing. And so we have these nucleotide sequences. So I showed you, fruit fly DNA is like 14% variable. That is, 14% of the genes are showing multiple alleles in a gene. But then if you look at it from the nucleotide way, that just means 1% of the nucleotides are different. So it's hardly anything that way. And so remember how they always say, oh, humans and chimps are like 99% the same, right? Well, humans and chimps are obviously vastly different organisms. We have lots in common, but we're considerably different from one another. And that just shows you that just that little bit, 1% of the DNA, that's enough to change a lot of genes, right? Okay. And then it also says, oh yeah, there's another way you could do it. You could look at it by chromosomes. So this is kind of a historical thing, just saying, oh look, here's genetic variation that somebody studied by looking at chromosome structure. And it's just showing you it's mapped out and pointing out to you that geographic isolation can lead to genetically variable populations. This population of mice over there on the west side is considerably genetically different than this population over here. And there's a big mountain range there that's preventing them from getting to each other. Then it talked about clines, and clines are another way that we find genetic variation in population, that there's some sort of matching of the genetics to the environment. And so the idea of a cline was, basically you go on sort of a march, like you go on a march up and over the Sierra Nevada, and you look at those yarrow plants that I talked to you about in the lecture, where the plants get shorter and shorter and shorter as they get up into colder and shorter growing seasons and windier places, until they get to the very top where they stop growing. And they're very short there. They're covered with snow most of the year. Big winds come and blow them over and stuff. They need to sprout up, flower, and pollinate each other and make some seeds for the next generation to come quick as can be. So they've got a real short growing season. Whereas those guys down there in the foothills of the Sierra Nevada that are by water and stuff, those guys are making big, tall, 2.5 foot plants with big, luxurious flowers, and they're spreading their seeds far and stuff. These guys, Keck, Kloss, and Heisey, took those plants and they transplanted them to see what would happen. And they saw that, what do you know? The tall plants from down low try to grow to be tall plants up above. They get smashed by the wind and all kinds of disaster happens, but they try to do it. And the short plants from above, when they get transplanted down to this ideal place in the foothills, what do you know? They stay short plants, right? So there was a genetic variation. So it's another way of showing that genetic variation exists in populations. That's what a cline is. It's showing you some sort of pattern of genetic variation. This graph here, the Momonchoks, may be a little easier to understand. It's just going along the east coast of the United States. And all the idea is that there's a biochemical allele, something called LDH, lactose dehydrogenase, and lactose dehydrogenase, this particular one. One form of the allele is found up in Maine, where the waters are near freezing, and a completely different form, is found down near Georgia, where the waters are very comfortable to swim in. And so there's just a variation over that spatial distance. And that's a cline. Okay?
Like an incline or a decline, I guess?
Hmm?
Like an incline or decline,
No, so it doesn't really use that. An incline is go up a hill, right? And
Yeah.
decline is to go down that hill. It's not really that. It's just a climb. It's just sort of some sort of straight line. It could be from the southwest to the northeast, or it could be north to south, or it could be whatever direction.
Oh, okay. I like that.
Okay. So that's all telling you, hey, genetic variation is out there. That's all this was really. That was the big message that this was telling you. Populations are genetically variable. Okay. And then we stopped and said, okay, well, wait, maybe we better talk about what populations are. And so I talked to you about these caribou in Alaska, the porcupine herd and the 40-mile herd those were two populations, but they overlapped each other, but they weren't the same population because one herd bred in one location and another herd bred in another location. And so to be a population, you have to be interbreeding, right? And so these guys weren't really interbreeding. I mean, every once in a while there's gene flow from one population to another, but they're not really interbreeding. They breed in different places, and therefore there are two populations there. Is that all right?
Yeah.
Okay. So then I mentioned to you that there's this thing called Hardy-Weinberg. This is what we're going to do on Monday because we can't have the computers. And on Monday, we'll study about Hardy-Weinberg. And Hardy-Weinberg is kind of like a control in an experiment, something to compare to. And so it's a way of looking at the genetics of a population, starting with Mendel's rules and expanding it, and then saying, okay, so this will tell us what this population would be like if there's no evolution involved at all. This is how many of this genotype and this genotype and this genotype we're going to have, and then if we go and measure that population, if we measure the biochemistry of that population, like we did with the fish, we measure all these individuals, and we find out that the numbers are different, then we know evolution really is happening in that population. Okay, so that'll become clear to you on Monday. We'll spend time on that on Monday. I don't want to talk about it anymore. Okay. And then lastly, then I got into, so there are multiple causes of evolution. We talked about natural selection a little bit. We're going to talk about it again, as it turns out. But we talked about it a little bit, but I wanted to talk to you about also genetic drift and gene flow. And gene flow, I really have talked to you guys about the whole idea of how San Jose has changed is my perfect example of gene flow. And I'm going to talk to you about a fabulous example that I guess I didn't get out of this textbook. Yeah, I got it out of a different book. I'm going to talk to you about another fabulous example of gene flow. Just checking
The
right to see.
Pennsylvania one.
It has to do with these heavy metal tolerant grasses in these mining towns of Arizona and some other places where they just have all this copper metals, tail slags and around and stuff. And so you can see some gene flow and what's really happening there in natural things. So we'll talk about gene flow on Monday. But we talked about genetic drift. And genetic drift is thought after natural selection to be probably the second most important process for most populations. So genetic drift, we think, has a real role in changing populations over time. And the first thing you should know is it's a random process. Okay, so it's random, and it roughly approximates the idea of sampling error in a poll. So again, I gave you guys the example, what's the best radio station in San Jose? And I ask one person and I pretend I know the answer. Well, maybe not, because maybe nobody else agrees. I ask two people, I still don't really know the answer. I've just got two names of radio stations, right? I ask three people and one of them agrees with that. Is that enough to stop? No. I've got to survey more, right? To get to the truth, you got to survey more. And the idea with gene flow, with genetic drift, is roughly the same thing, in that what's going to happen is populations are going to change based on the fact that the population numbers are going to be really, really small. Okay, so let me give you the two examples. The first one's called founder's effect, like the founding fathers. And so founder's effect means that you got a population over here, they're doing great. In fact, they're doing so great, there's not room for everybody. And so somebody gets up and sails and moves over here. Okay? Well, if the people that, or individuals, whatever organism they are, if those individuals perfectly represent the genetics of these guys, that is, if we get a perfect sample, then we're going to create, in a similar environment, a whole new population that's basically the same as this population, right? But that so seldom happens. What often happens then is a few individuals are going to come, and those guys have, you know, a little subset of the genetics that was represented here, and it's not completely representative genetics. And so now they come over here and they're very different. So my example to you was with this population from the southeastern corner of Germany of people that came over and settled in Lancaster County, Pennsylvania, to make the Amish or the Pennsylvania Dutch. These German people who came over happened to disproportionately highly bring with them that polydactyly thing where extra fingers and toes are there, and they happen to bring this very genetic form of depression that comes along, adult onset depression. And so the people in Lancaster County showed those two traits in much higher proportions than they did over in Germany at all. And so there's a genetic difference between the populations, and that's the founder effect. Does that make sense? Okay. Then the other one has to do with when you've got a very large population and some sort of catastrophe happens. In the example I gave you of marine mammals, the catastrophe was humans happened. And humans just said, oh man, we want to make hats out of your fur. And they just started indiscriminately killing all these animals. And so, you know, we go from, in the case of elephant seals, we go from like 2 million elephant seals down to 2,000 that are left on some island off of Baja, California. And that was it. So they were almost completely extinct. Hi, that's right, come on in, don't worry, I've lived. And so in that population crash, a huge number of alleles were lost. And that's where I showed you this figure here. And this figure, you know, this is how they call it the bottleneck. So these geneticists that were working all this stuff out, they had all these crazy ideas of how they could make people understand. And one kind of their genetics is called beanbag genetics, and this particular one's bottleneck, and it's from this metaphor right here. So they're just showing you, hey, look, if only a few individuals survive, they're not necessarily going to have a pure representation of the alleles that took hundreds of thousands to millions of years to make, right? If only a few survive by chance, and heck, look at what could happen. We could lose the yellow ones entirely, and we could switch from about 50-50 between the yellow and the blues to, you know, 80% blue and 20% yellow, or whatever it is over here, something like that, right? And so, again, it's just loss of genetic variation. And then this next example is one with the prairie chickens. I don't know if I made this point as clearly as I meant to, but I was trying to tell you that there are consequences to losing that genetic variation. And those consequences are reflected very nicely here in that what it's showing you is the fertility of those prairie chickens has gone way, way, way down, where genetic drift has caused a huge loss of alleles. And so the fertility of these guys where there's no bottleneck effect and no genetic drift remains about 99% of the eggs, 96% of the eggs are hatching, 93% in Illinois. But then now we have all this terrible bottlenecking effect in a couple populations in Illinois, and 63 years later, their percent of eggs hatching are less than 50%. So they're missing the genetic variation that helps them to deal with the environment, that helps them to stay healthy. And so loss of alleles is a bad thing to organisms, unless it's somehow guided by natural selection so that you only lose the ones that aren't helpful in your environment. Does that make sense?
Yeah.
Okay. I think that's as far as we are. Yeah, because we have to talk about this. We have to talk about natural selection next, and then talk about good genes and adaptations. Yeah. So we are halfway through this chapter, or maybe a little bit more, and then we'll have one last chapter to go, and then we'll be finished.
It'll be chapter 23, right? Speciation.
Yeah, speciation. So we're gonna talk about speciation as well. So that's what we'll do. So I'll finish this chapter. Let me just look.
Mundane start there.
So I have to talk about this, which is pretty straightforward.